So now we are on stage, right? Yeah, we are live. Hi, everyone. Hopefully you can see us, you can hear us. Please let us know if you can't or that you can see us. We are very glad to be here today. Again, I will mute some of you because, yeah, let me just. We are very glad to be here today with Luis Puelles, with Paul Cisek, and Georg Striedter, who join us for this last session of the reading club. We cannot think of a better way of wrapping up this series. We started with him, and he accepted the invitation to wrap up the reading club, and we are very thankful for that. He will actually give a presentation summarizing the last chapter and also summarizing their ideas when they wrote the book. So what I will do is I have a very short introduction. I see you can hear us. So I will share my screen and I have a very short introduction, and then I will let George present. So I'm in presenting mode. I hope if you cannot see what I'm showing, let me know. Otherwise, I will go on. This is a 5-slide presentation, but just to let you know that as I already said, this is the last chapter. This is the patterns and principles chapter in which we will discuss everything we have been discussing already, but trying to come up with these patterns and principles that characterize evolution. These are all the chapters that we have already discussed. We have learned a lot. We hope you have as well. We have also enjoyed a lot and hope you have enjoyed the sessions as well. And as I said, we have George today to summarize the chapter and also tell us something about how they wanted, why they did the book the way they did it, and what they were hoping for when they wrote it. Just in case you don't know us, we have Luis Puelles and Paul Cisek, who are always with us, making the comments, giving the interesting perspective. And we are the Braining Club. This is Alex, Raquel, and myself, who are organizing this event, and some others if you want to follow us in Twitter. And we need to acknowledge, especially Worldwide Neuro, because they let us use their platform throughout the whole reading club, Barxing community in Barcelona, and Instituto de Neurociencias in Alicante. And we, of course, this last chapter wants to thank all of you for being there in all these sessions, making questions, making comments. And again, we have enjoyed this a lot. Just let me remind you how to participate in case you don't know, because today we have a lot of time to discuss. So the more questions you make, the more we can discuss. You can make comments, you can share references that you think are relevant, or comment on what we are saying on the stage, here on the chat. And you can also ask questions in the Ask a Question button, or you can vote for questions that have been made, so we can know which questions are more interesting for you, the audience. So basically, this is my introduction. I will let George share his screen and tell us. I think I'm not sharing it anymore. So George, whenever you want. Yeah, we're seeing your desktop.
Can you see my screen now?
We're seeing the whole desktop. We are not seeing the presentation.
No.
We are seeing actually ourselves in the crowdcast. Maybe you didn't share the window you wanted to share, or
I'll do this one more time. We tried this yesterday and it worked yesterday.
It's always like that with crowdcast.
Does it work now?
No, we're seeing your desktop and the Crowdcast window. Are you selecting to share the whole? Okay, now we're seeing the mountain in the desktop. But we don't see at all the presentation, so it might be.
Yeah, I know. I'm working on it. Oh.
Okay.
Always something. I just restarted everything. I'm gonna share the screen one more time.
Mm-hmm.
And this is the window I want to share.
Okay, now we see the presentation. If you go in presentation mode or just maximize. Yeah, if you maximize it.
Yeah, I'm working on it.
The window.
How are we doing?
Right now we see the window, but okay, now it's perfect. Can you go one slide forward to see if, to make sure that we can follow? Are you moving?
Yeah.
It's not moving yet. Yeah, now it moved. Maybe it's just there is some delay.
It's just very slow, I think.
Yeah. Yeah, but
okay, I'm just going to start.
Yeah, go ahead.
Let me know if there's a problem.
Sure.
So thank you for having me on here. This whole thing has been, I don't know if you can imagine, but for a book author, there's like nothing greater than having this many bright people discuss something you wrote for this long. It's a little scary, but I'm very, very honored. So what I'm going to do today is summarize this chapter, but I'm going to try to do a very quick summary because I'm mostly interested in having a discussion. So we're thinking it should take maybe 35 minutes, something like that. And instead of just giving us pure summary, I'm going to add a little bit of personal context, sort of how I felt about some of the decisions we made and why we made them. And I just want to be very clear, these are my views. I didn't check this talk with Glenn Northcutt, my co-author. He obviously was a huge influence in writing the book, but I will just speak for myself. And if you have questions afterwards in the future, just send me email. I always love to get questions from people. So in terms of background and context, many of you may know that Rudolf Neuenhuis and his collaborators in 1998 wrote an enormous book. It's that 2,250 pages in 3 volumes. And it's an amazing book. It really describes the brains of vertebrates, all the different groups in amazing detail. And this paragraph here that I'm quoting came right at the end, and it really struck me as something that stood out for being, in a way, very honest about how he felt having done all of this work. And he basically says, here, I'm not going to read the whole quote, but he says that it would have been nice to find some clever and subtle principle that made sense of all of these different vertebrate brains, to reveal some kind of secret organization. But instead, he was feeling, that the whole thing is just so complex, and each box, like if you think of a brain in each box, and you're surrounded by all these different boxes, each brain has a distinct identity. So what he was really saying, in my mind, is that he saw and appreciated this enormous complexity and diversity, but he was looking for a pattern. He was looking for order in all this chaos. And I think that's what we're all trying to do. We're trying to find a pattern, when you call it a Bauplan or some organizing principle or some scaling laws. We're looking for principles in there. But once you have the principles, you're sort of still left with a lot of exceptions. That's kind of how biology works. To almost any rule and principle, you find some things that don't fit. And I think I'm just fascinated by that tension between order and disorder, looking for similarities, appreciating differences. And that's a very old idea. So I just changed slides. Let me know if it doesn't change. But I found this amazing quote from Francis Bacon, 1620, where he was already talking about how some people, some scientists, but all kinds of people prefer to look at the similarities in things, try to find the similarities, look for order in things. And other people are more struck with the differences between things. And what I really liked about this quote is how he says that each of them readily falls into excess. And that's a lesson I think we should all take, including myself, that we should not overemphasize the differences or the similarities, but think about them both. And I'll come back to this at the end. I have a second quote here that I discovered very recently from C.J. Herrick, who, as many of you may know, is the founder of the Journal of Comparative Neurology, and spent much of his life looking for general patterns among vertebrate brains, trying to synthesize all this information. And as Luis would probably say, with mixed success. But what I thought was really interesting about this quote is that he talks about how after you've looked for these similarities, these general patterns of organization, it's time perhaps to look for the things that have changed in vertebrate brains, for the differences. But that is a more difficult task. And I feel like that is still true today, that if you want to talk about similarities between, different vertebrate brains, that's relatively easy to explain and even publish because people accept, it's inherited from a common ancestor. That's why you have the similarities, and there's not much to explain. If, on the other hand, you want to describe something that's different, you really need to prove that it's really different. It's not just some, just looks, technical difficulties or something like that. And then you need to explain the differences. You need to say, well, why does this organism differ from that one? And then you have to prove selective pressures and adaptations and functions, and it becomes much more difficult to tell those stories. So I think we have an inherent sort of bias in our literature towards similarities just because it's easier to study those and explain those and understand those. And so I thought that was very interesting about C.J. Herrick. So now I'm going to just go through these sections a little bit for summary. I'm not really going to talk about the first section because you've already discussed the other chapters, and really all I do there is sort of concisely summarize the preceding chapters. So what I want to do is talk a little bit about some of these larger patterns of evolutionary change. And really, if you think about it, what I'm trying to do here, what we were trying to do here is not just recognize the similarities, but realize that there are differences, say from the Bauplan, or differences between species and along other dimensions. And then say, are there patterns in those, in those differences? How do we explain those differences? So I think here are just a couple of things that I pulled out that I think are interesting. This top graph here shows the number of tetrapod families. So it's a measure of diversity, really. How many different families of tetrapods were there starting in the Carboniferous and going all the way to today. And I've mapped on here some of the major extinction events. And this kind of surprised me because I always thought after major extinction events, you sort of slowly rebuild what you had before and the diversity would stay roughly the same. But you can see that diversity, at least in tetrapods, has really increased dramatically despite all of these extinctions. And what does that mean? To my mind, all it means is that evolution by natural selection is an enormous driver of diversity. It is really powerful. It really creates diversity over time. And if for no other reason, that would make diversity interesting, right, to explain, because it is part of evolution. I like to say that Darwin didn't just talk about things being inherited from a common ancestor. He talked about descent with modification. And that's really what I'm most interested in, is how do you account for the modification? The other thing that I think I learned from writing this book, and I learned a lot from Glenn because he was really good about understanding sort of fossil prehistoric fishes and other animals. And I didn't know much of anything about that before we started collaborating. So here's a plot of basically a measure of diversity. And you can see that placoderms, these are these giant armored large fishes, often very large, that sort of dominated the aquatic environment during the Devonian. They did really well during the Devonian and then became extinct. And the cartilaginous and bony fishes, around the same time, became, flourished and became more diverse. So there was a radiation of these bony fishes and cartilaginous fishes. And that's an interesting pattern. And you can ask, how do we explain that? You might be tempted to say, well, the bony fishes and the cartilaginous fishes sort of outcompeted the placoderms. Like they were competing with one another, and the bony and cartilaginous fishes were the winners. But if you want to make that argument, you need to show that they lived together in the same place at the same time, competed for the same resources, and that probably wasn't the case. It's probably better to think of the placoderms having gone extinct, and that opened up some new opportunities for bony and cartilaginous fishes to evolve into, if you will, to either take over some niches that were now empty or to create new niches. And so it's just an example of, you find these sort of patterns in the fossil record. Explaining them can be a little tricky, but I think it's still interesting and worth trying to do. Yeah, and here, just this little thing about all those fishes. You might notice that in this chapter, there's a lot of information about fishes, because at some point, Glenn and I realized that we had focused a lot of the preceding chapters on sort of the lineage leading to humans, and we sort of ignored this other huge lineage leading to goldfishes, right? So there are a lot of fishes, and so this chapter is sort of a little tilted towards them. Another really interesting pattern in the fossil record is this measure, is this issue of body size. So this is sometimes known as Cope's rule. There was a Cope, I forget his first name, studied mostly, I think, mammal fossils, and noticed that over time, the fossils tended, on average, to get larger and larger. This graph is from someone called Noel Heim, who looked at marine animals and found essentially the same pattern, that over time, marine animals became much more significantly larger. And he was nice enough for the book. I contacted him and I asked if he could break out the chordates as a separate graph for me. So he actually created this graph specifically for the book. And you can, because we were interested in chordates, not all kinds of other marine animals. And what's important here is that it's not just the mean, the average size, body size that increased over evolutionary time. It's also the minimum, right? So because you could imagine an argument that, well, you started with really small animals and then you just had random evolution, but you can't get smaller than very small. And so you just basically increase the variation on the upper limit, and that would increase the mean body size. But it actually seems to be the case that both the mean and the minimum body size increase. And that implies some sort of driver towards increased body size, maybe some selective factor towards larger body size. It's a little difficult to know exactly why that worked, but I think it's an important point that body size tended to increase for a reason. We don't know exactly what that reason is, but. Okay, so body size. What about brain size? Right? So we're spending a lot of time in this chapter talking about the evolution of brain size, independently of body size. And here the emphasis on fishes, again, in this case, cartilaginous fishes. And here's relative brain size along the axis, and all the black ones are basically negative residuals, where the animals have smaller than expected brain sizes when you consider all of these cartilaginous fishes together, and the red bars are positive residuals. So you can see that there are some cartilaginous fishes with just enormous brains. So I just love the devil rays and the manta rays. I've seen some of these brains in buckets at Glenn's lab. They've always fascinated me. They have this enormous telencephalon, enormous cerebellum. And you really got to wonder, what are they doing with these brains? We don't really know. We know they're fairly social animals, but we don't know much more. Some other cartilaginous fishes, in contrast, have really small brains with very small telencephalon, relatively small cerebellum. So there's a lot of variation. The important point here is that the increases in brain size have happened multiple times independently. So it's pretty clear that these animals here, which include the hammerhead sharks, for example, increase their relative brain size independently of these guys. And this is one of the general patterns we observe, that there are a lot of independent increases in brain size. Yes, there are some decreases in relative brain size, but they, from everything I can tell, they are not as frequent as the increases. We can talk about that. So another example are the cyclostomes, or some people call the jawless vertebrates. So these are your hagfishes over here, and the lampreys over here, the various species. There's really nice work done by, I forget his name, Salas, I forget his first name. And you can see that some lamprey, we'll start with hagfishes. So the hagfishes have these very surprisingly large brains, certainly large relative to lampreys, to most lampreys. And here's a section through the telencephalon. I had a good friend when I was an undergraduate, Helmut Wicht. He was a postdoc in Glenn's lab. He was looking for where the ventricle is so you could make out some sort of, homologies for the telencephalon, and it was very difficult. For now, all I'll say is you can clearly see some laminar organization here in these hagfish pallium. We still can have arguments about what's dorsal, medial, lateral, ventral pallium in these brains, or even, thalamic eminence. Not at all clear, or certainly debatable. But what I want to point out is that these are not really primitive brains, right? These are very strange, very derived, quite large brains. So if you're looking for a primitive fish, this hagfish is probably not the one to use. Lampreys, slightly different story, but again, I will come back to lampreys later. But first of all, there's some significant variation within lampreys. Some are parasitic, some are so-called river lampreys that are non-parasitic. I think overall, when Salas looked, he's concluded that the parasitic ones tend to have somewhat smaller brains, but you look at the pattern here, and it's not at all clear that only the non-parasitic ones have large brains. So here's the river lamprey, for example, is parasitic and has a fairly large brain. Why is there this variation? And are lamprey brains really primitive brains? I think it's certainly a good question. But the hagfish and lampreys have diverged from one another for, I think I looked it up, 400 million years, something like that. I'm bad with dates. And they obviously have evolved separately from the jawed vertebrates for an equal amount of time. Yeah, more than 400 million years. It's good to have notes, but then you have to look at them. Okay. So evolution of brain region size, right? Because some people rightfully argue, like, don't focus just on brain size. It's easy to measure, but what does it really mean? And maybe you should look at neuron numbers. Maybe you should look at individual brain regions, and those are certainly all valid points. I have long been intrigued by this observation that Barbara Finlay and Richard Darlington, and there's some precursors there, but I credit them for making this key observation that for a lot of animals, a lot of mammals initially, when you plot the size of individual brain regions against total brain mass, you get these predictable scaling relationships. That is, that as total brain size goes up, the size of individual brain regions goes up predictably. Something more than 90% of the variance is explained just by total brain mass. even more interesting is that when you chart the if you do these kinds of plots for different brain regions, you see that the slopes, the scaling slopes, are different, but for different regions, and they tend to be highest for the telencephalon and lowest for the medulla. And what that means is that as you go to larger and larger brains, it's especially the telencephalon, diencephalon, the forebrain, and the cerebellum that get enlarged, not so much the other regions. And you saw that with the manta ray. I mean, that's a pretty big forebrain. And in fact, these data here are for cartilaginous fishes. So you can see this general scaling rule in both cartilaginous fishes and mammals. Of course, there are exceptions, and a lot of people like to say, and I'm included in that we like to call those mosaic evolution, where you have individual brain regions that sort of go against this scaling pattern. And I think a really good example are the electric lobes of electric rays down in the medulla. They are just hugely hypertrophied, and there is no way that a simple scaling law could explain that. So when you try to understand the developmental mechanisms for why do these scaling rules exist, I wish we had better answers, I think. I still am not totally sure why developmentally, you can explain this scaling law by reference to neurogenesis, timing, schedules, and so forth. But why? I think there's still some important questions that remain to be answered. And for mosaic evolution, I think it's fairly easy to say, well, you just have more neurogenesis or less cell death or more protracted neurogenesis. But I still, you're left with a lot of sort of individual explanations. I would like to see it's maybe more comprehensive explanation. I don't know. How am I doing time-wise? Let me check. 20 minutes, okay. This chapter deals a lot with fishes. So I think it's interesting that when you look at the ray-finned fishes, and here's a brain of a triggerfish on the bottom, and a jack, which is a very fast-swimming ocean fish. Both of them have relatively big brains for teleost fishes, but they've done it in different ways. So the triggerfish have a very large telencephalon, and these jacks have a very large optic tectum. And so I think this is I'm using this as an example to suggest that maybe among teleost fishes, the concerted evolution scaling rules that I just talked about, maybe not as tight. There may be much more variance. Of course, it could be that within triggerfishes or within a larger group of lineage of fishes that they belong to, that you might still have a concerted scaling law. But when you compare these relatively distant relatives, they certainly have found different ways to enlarge their brain. The other thing I want to point out is that when you look at animals with, say, a large telencephalon relative to, say, a zebrafish, which has a fairly small telencephalon, it's not uncommon to find more subdivisions and often laminar organization in these relatively large brain regions, whether it's in the telencephalon. It's certainly true of the vagal lobe in cyprinid teleosts, that when you get these really enlarged structures, you often have more subdivisions, layered organization. And I think those are really interesting differences. By the way, we don't really know what's going on in this telencephalon and why the lamination. I think it's very interesting. Okay, now to something a little bit maybe more novel, a little more controversial. Homologies in the pallium. So Glenn and I spent a lot of time thinking about that. Here's a transverse section to the telencephalon of a teleost fish. This is from an old paper where they suggested, first of all, in a telencephalon of fishes, you have the structures are named by a terminology that I believe it was Nuenois who came up with it. I know it was Nuenois. That is not named, hippocampus, neocortex, amygdala, and so forth. Instead, they just named it dorsal medial, dorsal dorsal, dorsal lateral, dorsal central, and sort of a topographic nomenclature that I think was pretty wise, not to sort of name things for assumed homologies that may later turn out to be incorrect and then force you to rename things. When people started looking at the potential homologies, like what is this homologous to in this region, homologous to in a bird or amphibian, people often, looked towards connectivity, and that's certainly something I did in my dissertation. My dissertation was on basically the forebrain of various teleosts. I spent a lot of hours trying to compare connectivity diagrams and find homologous patterns, and I couldn't do it. In the end, I said, this does not work. Some people have said that it does work. So here in their paper, they suggested that the intracortical circuitry of mammals can be found in the telencephalon teleosts, but the structures are not arranged laminar. Instead, these cell types are distributed over different telencephalic areas. Some of you may recognize a shade of Harvey Carton's thinking in here, but my main point here is just to say that according to my thinking, you can try to find this kind of similarity, but you have to ignore a lot of differences to get there, and I was just never willing to do that. Nowadays, I mean, this hypothesis really hasn't gained any traction. I'm just pulling it out to make that point. Nowadays, when people are talking about the homologies of, say, the neocortex in teleost fishes, they usually focus on these two structures, DD and DC, because topologically they seem to originate from a portion of the ventricular zone up here that is topologically dorsal, and DC has long output connections that might be similar to, or are similar to some neocortical areas. When Glenn and I started thinking about, okay, how do we write about this? We thought, we shouldn't be just comparing teleosts. So here, teleosts on this diagram. We shouldn't just be comparing teleosts to tetrapods. We should look at some of the primitive clades or basal clades of the ray fin fishes. So teleosts are one group. Their sister group are these gars and bowfins. Next closest relatives are sturgeons and their relatives. And then we have Polypterus. And when you look at, say, Polypterus, they have a large forebrain, but the pallium is very, very simple. I'll show you a picture in a minute. Similarly with sturgeons, they have a very small telencephalon. So we said if instead of just comparing teleosts to tetrapods, we should first compare teleosts to sturgeons and Polypterus and see what we got. And here are some diagrams through the left hemisphere of a Polypterus, a sturgeon, and a teleost. And here's these structures I just talked about as being possible homologs of neocortex, or let's just call them maybe dorsal pallium. This would be, the candidate for dorsal pallium. When Glenn, and I have to say, Glenn did most of this work. I did some work on Polypterus when I was a student. A lot of this is unpublished work by Glenn. He spent a lot of time looking for DD and DC in sturgeons and Polypterus. And in Polypterus, he basically couldn't find either one of them. In sturgeons, you could find some DC. He did a lot of connectivity studies in sturgeons, and certainly on connectional criteria, there wasn't much of a DC that he could identify. So that then led to the argument that, okay, how can these two structures, how can they be homologous to neocortex if Polypterus and sturgeons don't have them? That would be a fairly unparsimonious argument. You'd have to say, well, these species lost those structures, and instead we think it's more parsimonious to say that maybe the last common ancestor of these guys didn't have DD and DC, and these structures are new with teleosts. And if that's true, then how can the DD and DC of teleosts be homologous to, say, the dorsal cortex of reptiles or the neocortex of mammals? At least it's something that one should question. And so we then started thinking about lungfishes and amphibians, which are, again, intermediate groups between teleosts and reptiles. And in lungfishes, their pallium looks very much in a way like the pallium of Polypterus, except, of course, it's not everted. And I assume you've talked about eversion versus evagination. But basically, you could find divisions within the lateral pallium and divisions within the medial pallium, but to both Glenn and me, it looked like there wasn't really anything here that would qualify for dorsal pallium. And I should say, neither Glenn and I are gene jocks, I suppose. I mean it in a positive way. But when we read the literature there, we didn't find any really compelling markers for dorsal pallium. I think that is one of the issues that we'll talk about in a minute, how that can be resolved. But anyway, so it looked like to us like there was really no dorsal pallium to speak of in lungfishes. And then we went to amphibians, and there everyone pretty much has recognized the dorsal pallium historically. But when I was trying to draw it and try to find figures to illustrate the dorsal pallium, I was struck how different people drew the boundaries of this dorsal pallium very differently in different species, but sometimes even in the same species. And I just became really uncertain about, like, where is this mythical dorsal pallium? And it's a little frustrating in the book, maybe for readers, to realize that Striedter and Northcutt really couldn't make up their mind very well. They were very cautious. What exactly were they saying about the dorsal pallium? But we were genuinely surprised at our failure to be convinced by a dorsal pallium in amphibians. And that's why a little bit of that confusion. I tried to clean that up in a later paper. But really, we were being speculative. And it was really amazing to me personally to see the recent work by Maria Tosques, where she did single-cell RNA-seq in salamanders. And here, this S stands for salamander. And basically what she did is she looked, for this analysis, she looked at turtles, salamanders, and mice, lots of neurons, looked at these gene expression patterns in individual neurons, subjected them to a big integrated cluster analysis. And then here she ordered the clusters by sort of where in the cortex they were are expressed. So you could see these clusters contain hippocampal neurons, these subicular neurons in mice, these entorhinal neurons, and neocortical neurons are here. So the big question then is, in the so-called dorsal pallium of salamanders, its cells, where do they cluster? What do they cluster with? Do they cluster with neocortex? Not so much, right? Instead, they seem to cluster much more with the part of the entorhinal cortex and part of the subiculum. So that was very much consistent with what Glenn and I had been thinking, even though we had no evidence. It seems to be the bane of my life that I have interesting ideas without much evidence. But I was very happy to see that we now have some actual, what I consider solid evidence. But there is a problem, and that is the lampreys. I don't want to spend too much time on this, but we already realized this during the writing of the book, that Grillner's lab has been working on lampreys, and here's a section through the telencephalon of a lamprey. Without going into the details here, but they describe a cortical area that gets thalamic input, has long descending connections, is responsive to retinal stimuli, and they're basically saying it's not just the basal ganglia. So Grillner has been saying this about basal ganglia for a long time. He's basically saying neocortex is also very much present in lampreys and is homologous to the neocortex of mammals. So since lampreys are way at the bottom, not the bottom, but say the left side of your vertebrate cladogram, and mammals on the other side, if they're really homologous, that means all the intermediate taxa ought to have a neocortex as well, unless they specifically lost it. And that's clearly runs counter to our hypothesis. So I am not going to say much more about that. I think we can... it will be interesting to see. I think sooner or later, somebody is going to do a single-cell RNA-seq analysis on lampreys, and I think that will give us some very interesting data. I think I'm wrapping up now, but I think the biggest problem that Glenn and I ran into when we were arguing that the dorsal pallium evolved at least twice independently in ray fin fishes, and then again in tetrapods, rather than being homologous across all vertebrates. The biggest problem we encountered is that people were like, wait, how can something like the dorsal pallium evolve from nothing, like new, be really new? You can't have anything new in evolution, is something we'd hear sometimes. But if you read the literature on evo-devo biology and you go outside of the brain literature, you look, for example, at the literature on jaw muscles. I mean, it's very clear that vertebrate jaws were a real innovation, that yes, branchial arches were involved, but the best papers I've read suggest that there were some important shifts of transcription factor activity into new areas or different areas from the ancestral pattern, and that gave you new interactions that led to these new structures. And I just like, some of you may know, I like parrots. Parrots have some jaw muscles that don't have homologs in other species, and people are sort of figuring out the molecular and cellular changes that have led to these novelties. I think it's in the evo-devo field at large, this is not very controversial that you can get new structures, not from nothing, but by recombining the expression of probably old genes, but in novel combinations. And I'm not going to go, for interest of time, I'm not going to go through our scenario here. I just, we suggested it's just a thought experiment. I'm not saying this is anything real, but we suggested that, the pallium is shaped by or specified, the different subdivisions are specified by morphogens that are secreted from the hem and the anti-hem. And I can imagine that in a large increase in the proliferation activity of this pallial zone would enlarge that area so that the morphogens that have a fixed sort of diffusion distance, right, wouldn't be able to reach, say, the central part of this pallium. And in this region, then you would have a new combination of transcription factors active, and maybe some new kinds of cells emerge. The very last thing I want to say is that what I'm really interested in actually is the evolution of connectivity. So if you're interested in the evolution of neural circuits, it's really hard to do if you're not sure about the homology of the regions that or the cell types that you're trying to homologize. But if you have some good homology criteria, so say for the olfactory bulb, right, is pretty much clear it's homologous across these species. Then you can ask, the connections of the olfactory bulb to different pallial areas. It seems pretty clear to me that there has been a big regression of the olfactory bulb projections. And I'm not going to go through the details of this diagram. I'm sure some of it is wrong. It's very difficult to draw these kinds of diagrams without making mistakes. But I think most people would admit that when you no longer use connections as your main criterion for homology, but you use things like gene expression patterns, then suddenly, you start to realize that a lot of connections have changed in vertebrate brains. And maybe I was influenced through my thesis work where I looked at the connections of a clearly homologous brain region in the forebrain of teleosts that were closely related to one another. And I still saw a lot of variation in the connectivity. And if you see that much variation in connectivity between closely related species, I can't imagine why you wouldn't see it when you look at very distantly related species like lampreys and lizards or something. In terms, bringing it back to the larger philosophy, what I think is very interesting is what Daniel Lehrman called the natural history orientation. And that's really the view that we took in this book. Daniel Lehrman was an animal psychologist, an American psychologist, and I think he was here with this quote, was mainly complaining about people like Skinner and Thorndike, who had a very overarching general theory of how learning and memory works, and weren't really interested in the specific variation of what animals learn. So that's why, for example, conditioned taste aversion didn't really fit into their scheme. And Daniel Lerman is saying, like, no, instead of starting from a big theoretical framework, start with the animals, start with the organisms, figure out, what are their particular conditions of life, and then see how they fit into any framework if they do, or maybe sometimes they don't. And what I really like, going back to this thing about some people preferring similarities and others preferring differences, I really think we ought to try to have, keep both in mind and have them illuminate one another. And with that, I want to finish. I just want to say maybe a little bit of advertising here, but this was a book I wrote right after this one, in which I tried to address the question of, like, who cares, right? Who cares about all these differences between different species and their natural history? Who cares about the history anyway? And my answer in this book is that if you're, there are many reasons to be interested in history, for example, but one reason is that if you're interested in biomedical research and you're using model organisms, a lot of times your translational efforts will fail because you didn't take into account some important species differences. And they may be minor, but when it comes to translation, minor differences often make a big difference. And I think that's an important lesson. So with that, I want to end. I want to make sure I can unshare my screen, but I'm at 40 minutes, and I think that's kind of what I promised.
Thanks, George. You'll have to switch on your camera. I switch all cameras off because
I'm working on it.
Because there were parts of the
I'm
slide
working on it. I don't know what happened.
Yeah, I'm trying to help you, but
if I disappear, I'll be right back. Here we are.
Okay, thanks a lot. This was great. I guess that we already have 3 questions, but I'm sure participants have many other questions, and maybe Luis or Paul want to comment on what you said. You are both muted, so if you want to say something, just unmute yourself. Okay. No, you are both muted.
Hi, I just wanted to say thanks, George, for writing the book. First of all, this has been a tremendous source of insights and a great place to start jumping into the literature, because your reference lists are fantastic. They are, and one of the things that actually I got to commend you on is that you're citing papers that came out a week before the book came out, and that quite surprised me. Like, you have that paper from Arendt's group about the Amphioxus, and that was like the day before your book came out, I think. So it's certainly a great resource. So anyway, that's the first thing I wanted to say, and then I'll pass it to others. It was a wonderful book.
I see all those reprint folders behind Luis, and I sometimes think that the kinds of books I've been working on, I couldn't do them without computers. And I have a PDF management system that has 12,000 PDFs in it, and I just, like, if I had still had the paper piles that I had when I was a grad student, that there was no way I could do any of this. So
you also have to have it organized in your brain, though. I mean, that's
a little bit.
That's the part that I find harder. Well, I could start it off, and I could start off with the question that I actually posted there in the question and answer, just because I wanted to type something in there, and that's really. I mean, there's a broader question. So one is there's a lot of question, of course, what's homologous and so it's not homologous. But for somebody like me who really wants to think about the function, the circuit is actually the thing I'm interested in too. And one of the sort of big picture stories that seems to be very consistent in evolution, in studies of evolution, is that things shifted to the telencephalon in mammals much more dramatically. And so, but I'd like to get a bit more on that. And particularly, I want to just jump right into that lamprey conundrum. That um, my As I read the literature, what I perceive often is that things we thought were new 50 years ago turn out to be old 5 years ago. A lot of maybe it's because people focus so much on similarities, as you say. Um, but Grillner's work on the lamprey is potentially very important because it shows in exquisite detail these projections from the lateral pallium in particular, and remarkable similarities too. 80% of the neurons are excitatory, organized in layers, not unlike what you find in amniotes and even mammals. But then we have, this giant gap between lamprey on one side and mammals and birds on the other. But is that gap there simply because people haven't studied enough of those intervening species, that we need somebody doing that kind of tracing and optogenetics on beach ears or on non-avian reptiles, and et cetera, et cetera? I mean, do you think that 50 years from now we will again have revised that, oh my, these actually are older than we thought? Or do you think that we'll be maybe less prone to assume similarities, and we'll realize that no, lamprey, that lineage came up with that,
Yeah,
later.
I don't know. I'll give a short answer. I think my sense is that in morphology generally, as people have filled in the intervening species, they have more often than not realized how much convergent evolution there has been, rather than, oh, amazingly conserved.
I see.
And so I don't know for the brain. I don't know, but I think as these phylogenetic trees, right? You need a good phylogenetic tree as they've become more robust, and as people have looked at more and more intervening species. You see, just like with us with these independent increases in brain size, you see things are popping up in multiple places on the tree, and I think you're going to see the same thing with some of these quote conserved features. But I agree with you that the more people look, the more they find similarities. But I think in part there's also this bias towards, they see differences as well. They just don't talk about them because it's, like I said, it's much harder to make a good story about the differences.
So let me ask you about some specific examples. So one of the things that, for example, I found interesting is that Glenn Northcutt, in his earlier work, talks about projections from the telencephalon of I think alligators, and talks about it much more in terms of a conserved view. But then, obviously, at this point, he and you have abandoned some of that, and so. How did that? Is it simply because he decided that this was yet another convergent thing in alligators, or?
So I have to admit, I don't know the details. I think you had raised that question before, and I looked up that paper. It's like a 1968 paper, I think.
Yes,
and
something like that.
And I think the projections are to the midbrain mostly. And when I talk about long descending projections, I tend to talk about projections that go lower than the midbrain.
Okay.
But I don't know. I mean, connectivity is hard, right? You need to confirm it. So
yeah, I think the problem is that it takes a lot of work, and a lot of people are drawn to, well, let's just do it in mice or zebrafish, right? And we have these little points that are so far apart, but somebody's need to do features, right?
I talked to Trygve Bakken yesterday from the Allen Institute, who is doing a lot of the single cell, sequencing and then cluster analysis. And I think, those guys are very close, I think, to being able to trace the connectivity of identified neurons where they know very clearly, what cell population they have. And when that becomes comparative, I mean, I agree at this point, it's mostly mice, but they're already finding differences or interesting things about the connectivity in different strains of mice, right? So I think that's where I'm.
Yeah, but how different though? How different? I mean,
major
track. Like for example,
different enough to make a behavioral difference, which is something I care about. If you don't care about that, then maybe not that different.
Yeah. Right. Yeah. Is this the barcoding approach? Is that what they call it?
Well, that's one approach. Yeah.
Yeah.
I think.
Anyway, all right. I'll mute myself so Luis can speak.
May I ask? I know you put the thing as a difference between people that like novelties in a brain evolution and people that like conservativeness. No. In that sense, probably I am your opposite. Also, I don't think that I don't like novelties, but it's okay. The question is, how would you say is the proportion in the things that happen in brain evolution that can be of very diverse types? How many times is introducing novelty, or how many times is just adapting what you had before to new conditions? How do you see the equilibrium between conserving and adding new novelties, which both things happens? I agree on that.
Right. I think I'd have to think about it more, but I suspect conservation is way more common. Novelty is rare. But I think novelty is important because, like, this was one of the main themes of our book, is that
it opens new pathways. No?
Exactly. You can do new things. It opens up new possibilities, and then that affects other aspects of your body and can drive evolutionary change in other things. So, and just like you acknowledge the importance of novelties, I would say you can't even start thinking of, you can't even identify novelties until you've seen the conserved pattern, right? No. So, right. I mean, otherwise everything is chaos. Everything is unconnected,
and actually, when in this sense you mention similarities, I disagree slightly, because conservation sometimes destroys also similarity and still is homology. So homology does not depend on similarity. It can be dissimilar homology.
Right. And this is something I've spent a lot of time worrying about, thinking about, to the point where I'm sick of thinking about it. But at what point do you say this is no longer homologous, right? Because say.
What about if you consider that novelty is simply added to homology? That homology stays, and then you add novelties and new possibilities to make things more complicated and more pathways opening. But I don't think that you can really lose homology anytime. I think that it may change, become less and less similar, but still it is there. Your head is like the head of a planarian, okay? Or not. Which is similar in many aspects, but it's your head, and he also has a head. Okay?
Yeah. I think
homology is there, but there's much novelty, of course.
Right. So I agree with you up to a point. So I think it's dangerous or unwise to say that, homology, as long as it's 80% similar, I won't call it new. But if it becomes only 40% similar, I start calling it new. That's not what I mean. What I mean is sometimes you have, an extra body part. You can account for all the ancestral parts. Your whole parts list is there, but you have a novel part. And now it's difficult to explain, where did that come from? And you can say, oh, it must be some old part that duplicated and diverged so much that it's new.
That's precisely the problem.
But you could also say maybe there's some novel interaction that happens, some novel, gene transcription factor interaction. So actually something new, a new type of cell or a new type of tissue can appear. I think that's possible, and that's what I'm most interested in.
Mhm.
Yeah, if I can jump in there, I think one of the problems we have in neuroscience, and I think it's here in Evo Devo as well, is that in order to express our ideas, we have to use definitions, right? And so we say, I define something called dorsal pallium, and I have certain criteria that are required to be met. And it also applies to the word new, right? We want to say whether it's something, either it's new or it's not new. But as you just pointed out, it's 80% the same, but the 20% is different. And so how do you know whether that's enough to justify calling it new, et cetera, et cetera? And I actually think that potentially the way to sort of avoid the grip that has on our those definitions have on our thinking is to do a lot more computational modeling, actually. So in computational neuroscience, very often certain debates disappear, right? Like there's a debate about the parietal cortex. Is it attention or is it intention? And then both groups will make computational models of their processes. And it turns out the models are actually very similar, right? They have similar dynamics, similar phenomena occur. And at some point, it just turns out that, well, you don't actually have to argue about what you call it. The computational model tells you all that you really want to know, right? So I think also likewise, perhaps if we had, and we're far from this, of course, right? Because we can't even model existing species, much less ones that died out, 500 million years ago. But if we had at least the theory of the functional, dynamics of a particular circuit, then I think we can have a better discussion about, well, what happens if these thalamic projections shift over here now? Then we have, I think, an idea about, all right, well, what's the implication of that? And so I think, in a sense, it's like that attitude of modeling the stuff might help us get around having to have these arguments even. And turns out that we actually, I think it's kind of wishful thinking because, again, I think we're very far from actually having decent models at this level. But I'm hopeful that, my grandchildren will.
I just briefly respond. I agree with everything you said. I'm worried sometimes when people get too focused on definitions, like this character, this trait, this is the essential criterion. If it doesn't have that, it isn't that character. So again, I was talking with back in yesterday about, they found a cluster of cells in one species, and they found it in the other species, but these were parvalbumin interneurons, and in one of the species, it didn't express parvalbumin. They don't have parvalbumin. They lost the parvalbumin. But in mice, we define them as the parvalbumin interneurons. But it's the same cell type. They just no longer express parvalbumin. So you have to be very careful about, using sort of essential criteria. So
it's essentially what Mayer said, right? You should get away from typological thinking.
Right. Don't become Aristotle.
So we had so Barbara had a related question.
Barbara. Yeah. Hi, Barbara.
Hello there. How's everybody? Can you hear me?
Yes.
Okay, great. So I think my question concerns kind of the relation between computational mechanisms and function, plus in contrast to developmental mechanisms and perhaps conservation. So when you started off, you were saying that it's hard to find explanations for diversity, but I've really found it in my own stuff to be just the opposite for just the reason you gave, because you don't have to account for this conserved function because it's just inherited. So it demands, but it was never explained in the animal it was inherited from. And so for a long time, and the way I thought about it, it got stuck in developmental conservation. It was there because it somehow had to be, because it was a sort of basal. But then after a while, that struck me as just bizarre for things to be conserved over the range of, ancestral time we were talking about. So then that turned it around. Now your problem is to come up with a reasonable functional explanation that transcends species differences, not is ignored by them. And so that's where you have to go to computation, I think. And development is not an explanation the way you put it's an explanation of how the size differences come about, but they're under the same, selection presumably as what produces the adult state, unless we want to talk about a specific case of a stuck in conserved thing, some kind of constraint. Let's put that aside for the moment. So I mean, it seems for like why forebrain should get larger, because of all the ways you can enlarge a brain. It's a permissive way for evolution. Suppose you have a set of cells that you have a long development, and by that virtue, they get relatively larger, and they are also, say, more permissive in the number of sensory systems they will allow to synapse there. Then bravo, there you've got a mechanism that is a computational advantage when you are in the position to be able to support extra brain, that you will be able to use it. I mean, I don't think we have to go to sort of always extreme species by species differences to come up with useful computational and functional explanations that transcend them all. That's So
I totally agree, and the way I put it, and maybe I've put, I was trying to condense. But I love Darwin's idea of looking for principles of variation. So the point is not to find just idiosyncrasies here and there, but to, once you've found some, try to, synthesize and explain them not on a one-by-one basis, but in terms of some general principles, whether you call them computational or not. I think,
right.
Yeah. So I totally agree.
But then it's always been, I think you were expressing some discomfort with the sort of name the area first and then figure out whether it's homologous or not according to some set of principles, or I'm probably misputting how you said that. But I mean, would it be better to, say, look at a large number of basal forms and say, okay, compared to all structures in the brain, which of those structures show the most variability from one animal to the next, and look at variability as the conserved thing, if you see what I mean, not the, so it would be a permissive set of tissues, say in the telencephalon or the cerebellum, that would be the informative thing, not their exact, nomenclature.
I did some work with Christine on bird brains, different bird species, and it's pretty clear that when you hatch, you might be a very altricial species, but when you hatch, certain things, parts of your brain have to be fully functional. It's okay for your forebrain to still be growing, and you can't really do anything with it for a while. And in the long run, then you reap benefits from having a more plastic forebrain at stages when you listen to your parents sing songs, and so there are ways to understand that. And I think you were clear on demonstrating that when these neurogenetic schedules, when you stretch them, you tend to enlarge the forebrain areas much more than say some of the hindbrain areas, and I think that I still would like to know sort of at the mechanistic level why that happens, but I think functionally it makes sense to me that you would be a very poor animal if your medulla took a really long time to mature, right? Because you can't grow, and that was an important insight that I read somewhere when I was thinking about all this stuff, that you can't prolong neurogenesis without delaying maturation. It's a very critical trade-off.
And also, it's computationally too. It's a very poor idea to make a required intermediate communication structure extremely large. It just wrecks the computer basically. So you often build these, side re-entrant kind of thing, so you can make use of information without slowing yourself down terminally, and you see that kind of thing emerging. I'll stop. So,
And the other thing I want to come back to that you said earlier that I think is really interesting, that in a way, conservation needs to be explained as well, right? We sort of
totally,
just,
yes.
you know, and very few people have written about that. You know, some people were trying to explain the phylotypic stage and why it is more constrained, the hourglass development model. But I've never fully bought those explanations. They never, you know, it always seemed like they could, you know, we need a little bit more to explain why there's so little variation in some things.
Yeah, that's a plea towards a developmental constraint sort of argument. That's not at all. So we got to skip that thing and go straight to what does the conserved thing permit, not what does it, like you just said.
And it'll probably be a little bit of both. I mean, constraints are real, but they're not everything. Yeah.
So I think Luis
I
wanted
had
to say
to
something.
mention just the general idea that this overall tendency to a greater size, the increase in size of the brain, maybe it has to do to just a general property of systems, of complex systems, that they are given thresholds of size or number of units that relate to the possibility of new properties to appear. Suddenly you have so many million circuits intervening in a perception or something, an item function. You cannot separate given categories, or you cannot give this or that treatment. So maybe that's why you see a slow increase the more intelligent the animals become, or the more capable to do things. On the other hand, I want also to put on the table the issue of the miniaturized brains, that we know some species develop smaller brains because they have not much to eat, apparently, according to what I read. I don't know, Georg, whether that is true any longer. Miniaturization, what's the cause? It's still lack of food, basically, but it's an adaptation to reduce food, or not. These are very small mice brains and thorax brains and so on. They have really diminutive brains, but they have everything. They have all the circuits and they have all the functions. So brains can function also being reduced secondarily. No? So that is also curious, no?
Yeah. I don't have a good answer to that. I think, I mean, one of the more, as I reread the chapter yesterday, and I thought, oh, this is kind of an interesting idea. I don't know if I really believe this anymore, but the idea that the pedomorphosis of early land vertebrates, you know, that they have relatively simplified, smaller brains. If you think of that maybe as an adaptation to a low oxygen, low predator load kind of environment, I think that's kind of interesting. That you know, I just haven't maybe thought about it enough. It's hard to prove these things, right?
There are many ways to the possible. What is possible? No? Not a single pathway. There are many ways in which you can survive.
I mean, I've always been amazed at, I think, Gerhard Roth, I think, did some of this work on these little salamanders, the miniaturized plethodontid salamanders, I think, and showing that, you know, their brain, when you first look at it, looks like it has very little differentiation. But when you look in the right way with the, say, connectivity, you see all this intricate patterns. That's fun.
Maybe we can move to the next question. That is by Esther Desfilis. I will invite Esther. Screen. Well, maybe she takes. I know. Actually, she cannot join apparently. So, yeah. Yeah, maybe she cannot join because she rejected the invitation. I can read the question. That is, one of the most controversial topics has been the evolution of the neocortex. Could this be due to the interest of researchers in highlighting the relevance of the species they work with as a model to study human brain disorders? Could it be that now that the amygdala has gained interest in relation to psychiatric disorders, the homology of neocortex will lose interest in favor of the amygdala? You wanna?
I like the amygdala. I'm tired sometimes of the neocortex, so I can sympathize. But you know, I think people, when it comes to trying to use a model species for to translate to humans, just because you're studying the homologue of the human amygdala, like Luis was saying, you can't expect it to be similar. Just because it's homologous doesn't mean it's going to be similar in all respects, and maybe especially not the respects that you need to have to make it translationally useful. That's my main problem. I spend a lot of time thinking about this with birds, right? Because I work on birds, and there are some parts of the enlarged bird forebrain are clearly homologous to the amygdala. Not all of it, obviously, but some of it. And I ask myself the question, so what does that mean? Does that tell me anything about how it works? Because does that mean now that I know this part is homologous to the mammalian amygdala? Can I transfer the knowledge from a mouse amygdala to the birds? And I've never really been able to do that very well. I've not been satisfied. Is it fear memory now suddenly, right? Because our definitions of what the amygdala does are not sophisticated enough at maybe the computational level to really transfer. I don't know. I'm babbling here, but what do you all think?
I think that we lack too much about knowledge of the amygdala to speak meaningfully about its functions, either in animals or in men. Because I in recent years have been looking at the amygdala in mouse and trying to think about it in general, and the impression is just that we are just at the very beginning of understanding how complex the amygdala is, even in a small mammal like the mouse. And of course, in humans and primates, it must be much more complicated, particularly its connectivity with the cortex and so on must be highly evolved. So that means that to derive conclusions and extrapolate functions and so on is, I feel that it's premature to think right now in this direction. So that is my feeling. So we have to wait 20 years or so, at least. That's what we recommend.
I think explanation affords a vacuum, and the one really useful thing that's happened is that the amygdala has finally been released from the grip of mammalian fear, and its really bizarre, limited definition that it got. But now looking at a number of different animals and not all mammals, something in that region of the brain, which is serves like a negative, not necessarily fear, but sort of an interrupt function, reconsider, put it in, consider and reconsider in the terms of viscerosensory information as well as sensory. I don't think it's the greatest idea to withhold a functional explanation just because you haven't proven it. It's to motivate and to get some better idea, and hopefully whatever you come up with is not going to have the stranglehold function that fear had for all those years about the amygdala. Yeah.
I think that's fair. At the starting point,
The whole literature on amygdala connections in rodents is based on a schemata, on an anatomical schema that contains only 10 nuclei, let's say, 10 main nuclei, okay? But we have found that there are at least 25. So what does it make of your conclusions about functions? If we find that if there are 25, then only 10 things have been studied, and we don't know how they relate one to another. So we need to study more and more until this thing gets cleared, because right now it's very confused. What is what is true, what is not. So the problem is that we are changing the paradigm of what is a schema, or what do we mean with amygdala. What do we mean with that? So it's changing right now. It's changing in these recent years. So we will need a number of years and a lot of research to clear out what is good or what we knew before and what needs to be added as novel knowledge or conclusions to what we had before. And then some of the old stuff will be false, simply. So that means that it's difficult to enter functionally the field right now. That's my impression, but you can do what you want. That is a risky area right now.
I think I understand what you're saying, Luis, and I don't disagree strongly, but I also worry, maybe this is a general issue for especially maybe the younger people in the group, that we have such techniques now to get, more and more data. We can, you can get 25 cell groups, you can get, the number of cell types in the human brain is exploding, and many of them are very rare, and we haven't even started thinking about the connection, or, soon we'll get connectivity data for all of them. And, there's a fire hose of data coming at us, even just for a single species. And at some point you go, how much more data do we want before we allow ourselves to sort of transition to the kinds of questions that we want to answer, say, about function?
Well, normally people have never waited, and people have gone on with functional interpretations, only that after 50 years it was found it was wrong. What happened with Herrick and his columns in the diencephalon? Were they functionally like the columns in the hindbrain? No, they were not. So it was wrong. The complete thing was wrong. But he went ahead with it, and people follow, and then later we have had to correct. So that's the normal history of neuroscience. That's sort of normal. But normally, function should be built on good anatomy. That is my point. If you can get good anatomy first, that is better. You do less risky stuff.
Yeah.
Okay, maybe
Esther in the chat.
We can move to the next question. I will invite, I think it's David Murphy. But in the meantime, Paul, if you want to add something, I don't know if you want to add something to the conversation.
Actually, my computer's performance is suddenly really bad. I might need to log in. I'll put it in strict.
Yeah, because
anatomy
your mic is not working.
Oh.
Yeah. If you want to log out and try to log in.
While we're dealing with these technical issues, I thought I might say something about the first question that was submitted on the Ask a Question thing about the brain being a thermodynamic organ, and does it dissipate heat at all? Is anything known about that? And I thought it was kind of it would be fun in part of the answer to say that was Aristotle's original proposed explanation for the convolutions of the human brain, that it was, had a radiator design that was ideal for dissipating heat. But I've never heard anything about the brain dissipating heat, but I know that there are, it's more important in a way to maintain a certain heat in, a brain at a certain temperature. And in that context, I think I once saw a mold of the rete mirabile. This is a network of very fine blood vessels around the brain in a manta ray. They have a beautiful, elaborate system that we don't really understand what these blood vessels are doing, but the hypothesis is that they help to basically warm the brain a little bit. These are cold-blooded animals, obviously, but so things like that are very interesting to me.
Yeah, there's an old paper by Dean Falk in early BBS, way earlier than I was, and she starts out with a quotation from her own car mechanic, where he kicks the radiator and says that the engine can't be any bigger than this thing. So, and then she reviews in that paper
Okay.
a lot of information about rather not the problem of heating the brain, but rather getting heat out of the brain and the particular things that are involved in dissipating a lot through the face, actually. And she talks, and turns out upright posture in giraffes or whatever is related to the kind of heat dissipation that is possible. So I haven't looked at that paper in ages, but it's a good paper. Interesting.
Maybe that's why I'm going bald. It's a heat dissipation thing.
Exactly. That's the reason for male pattern baldness, but not. But then
too much hot air, right?
Yes. Not much going on here. So.
Okay. Yes. So that was a question by Alejandro. I think he was asking about whether it was answered or not. So I will read David Murphy's question because he cannot join the stage. Is at some point in evolutionary history, animals began to pass knowledge down to dependents. For example, teaching certain hunting styles or using a particular tool. This is highest in humans, but it is present in other mammals and crows, if I'm correct. Can you identify a rise in brain structures that enable this externalization of knowledge? Did this arise more than once?
Wow.
Hi, Paul.
I don't know that anyone has looked at that systematically. I know a few examples, right? So it was already mentioned tool use, the transmission of tool use. I think, some nice evidence in dolphins that they pass on the use of sponges to collect food. But I don't know, so you can, I'm sure there are many papers on the evolution of culture, or at least a few, but the neural correlates of that, I don't think anyone would be brave enough to say anything about that. I don't know. And Barbara, do you know anything? No?
Yeah. I'd recommend you look at the Wayland stuff. So not so much in the brain part, but I think the distinction between sort of tool use and what is not is maybe too high up there. And so what he does, he looks at a lot of fish actually. And so this question of there is imitation learning in fish, not in the sense of like tutelage exactly, but just that they'll learn from the behavior of the crowd or of individuals. And then he talks about, okay, well then which individuals and you can show that just imitating everyone is worse than imitating no one. So then it pays you to get to follow an old one who at least is not died yet. Okay. And, so there's a whole bunch of different sort of levels of what an animal will pay attention to what end, that he does a way much better job of systematizing than I think anyone who starts from humans down does.
Okay. Who's this, Wayland?
Oh, Kevin Leyland.
Kevin Leyland. Oh,
yeah,
right.
Evo Devo, man,
Right.
Evo Devo website. And so, but his earliest stuff was all about cross species, who learns from whom and
Okay.
what, and not just this very kind of niche thing that niche in the sense of cutesy little thing, not
Right.
ecological niche that people.
Interesting. I was just discussing with someone about long distance migrants in birds.
Yeah, exactly.
There's some good evidence that some young birds are learning from the older ones that they migrate together with. But it's not that the older ones are teaching the younger ones. It's probably just that the younger ones are just learning from, they're just following.
They still exist, so they did it. So
yeah.
Yeah. So maybe we can move to the next question. I will invite Anastasia on the screen.
What about Paul's question?
Paul's questions?
He's here already.
But Paul's questions was answered, right? Paul?
He needs a microphone.
He's nodding.
Yes. My question.
Oh, damn.
Well, answer. An answer. Not tough.
Because your mic is not working great.
Mm-hmm.
Can you hear me?
Yeah, but it
breaks
has it breaks. Yeah, the sound breaks. Well, let's read Anastasia's question because she cannot join. Are you aware of any studies that compare sleep states in different brains across the evolutionary tree? I know there are different REM-like sleep states that are even observed in octopuses and even fruit flies. Do you have any hypothesis on at which point in time sleep originated and what could be its function?
I think Science had a whole volume dedicated to sleep a few months ago. I didn't read it, I didn't memorize it. But they were basically saying that periods of rest and activity are just really almost universal in life, that you pause transcription for a while and then you ramp it up again. But if the question is about sleep states, that's a much more nuanced question, right? So you can ask who has REM sleep, for example. And I'm not up on that literature. I know Gilles Laurent has done some really nice work with these lizards, where they have pretty good evidence, I think, for REM sleep in the lizards. But I'm not an expert on that.
Yeah.
I have a question, if I can Ben. And this one is actually for Barb. But George, I'd like to hear what you have to say about it. So one of the things that can't hear, huh?
Oh, it's got
Maybe
better.
now it seemed to work. Let's see. It's the mic.
Understand what the problem.
Maybe you can write the question in the ask a question button. And we can go for Kivshi question that says a fairly popular theory is that phenomenal consciousness or top-down control is based on the thalamocortical loop. In the book, you describe something close to these areas arising at the beginning of vertebrates with olfactory focused cortex and visual thalamic-like areas. Wondering if you have any thoughts on connecting the first theory with this evolutionary story. Consciousness.
Yeah. I love consciousness, when I have it. But I am very reluctant to speculate about the origins of it or the links to it. I'm not saying it can't be done. I was down the hall from Christoph Koch when I was a postdoc, so I know people are making good faith efforts to try to understand it. So I can't really say much. I think that this question of when did the thalamocortical loops evolve, I think is a more tractable question. And I haven't reviewed all of that recently, so I'm not sure I have full command of the data here. But I do think just because you have projections from the thalamus to the cortex doesn't mean you're going to have reciprocal connections back. Yeah. There might be a long circuitous way to get back to the thalamus, like in birds. There are some ways to get back to the thalamus, but that's not the same thing as a sort of true reciprocal connection that has special dynamics of its own. And who has studied the dynamics of thalamocortical processing in anything that's not a mammal anyway?
Can I make? I don't want to, but I have a paper that I absolutely love that everybody keeps ignoring, and that's Bjorn's paper on consciousness without cortex. And he makes what I think is an empirically based. I mean, there is virtually no direct evidence that says thalamocortical anything has anything to do with consciousness. You lose half your brain, front, back, side, and you're still conscious, right? It doesn't disappear with that, but the midbrain seems to be, is something that is highly conserved, has multisensory inputs, or, has a direct control of action, approach, and avoid, and integrates a whole lot of things. And there's all these little unusual things, like there's a whole bunch of unfortunate grad students who are anesthetized as part of their graduate career, and then they are imaging them and seeing what comes back. And then most people, tend to look at cortex because they believe that's where consciousness is, and in fact, it gets more synchronized when consciousness. But the very first thing comes on when the person when you're yelling, when you're sort of kicking them and saying, what's your name or something like that, it's the midbrain that's first up, and when that's on, the person can answer. So there are a whole bunch of things. So I really highly recommend Bjornberger, BBS, I think it's 09 or 07 rather, about consciousness without cortex, and it's a wholly empirical article from all the very old neurology literature as well as current stuff. And I don't know why this idea that somehow the cortex is critical got to be so enshrined, but there's really just no reason for it. I mean, I don't know. Stop. Rant ended. Okay.
No, I think you pointed me to that paper a long time ago, and I did read it. I can recommend it. Yeah, it's a good paper, and I think even when people talk about consciousness, to what extent are we really talking about attention, right? In which case, it makes sense that the midbrain would be involved. But so I get scared of these definitional questions. I just stay away.
Okay, so we had a question by Alex, actually, who is here. Alex?
Yeah, well, I have a small question. It's about cortical folding, not to do with head dissipation, though. But so in the book, you say that the last common ancestor for mammals probably had a small small cortex with no folds. And from the fossils that we have, it seems obvious that this is how it was, no? But in my lab, we think in a quite some different way, because we think that probably at least the developmental tools that allow for this great expansion and folding of the neocortex were already in the common ancestor, because we have girencephalic and mesencephalic species in all the main mammal lineages. And this cortical expansion happens developmentally in a very similar way, with the appearance of new or of the secondary germinal layers of new cell progenitor cell types, etc. So I was wondering, what do you have to say? And also what Luis said before about these miniaturized brains made me think about this, that maybe in some mammal species have had a secondary loss of these folds when they became smaller. Like, for example, could be the case of mice. So what do you think about that?
So I'm aware that there was an argument published, I forget his name now, but German, I think out of a German lab, that where, if you do a reconstruction of like all the lissencephalic, gyrencephalic species, you do get, you do the ancestral state reconstruction, you do get something that has folds. So I don't deny that. For me, the important point is that when you look at the correlation between brain size and folding, it seems pretty clear that when you get to very small brains, put it differently, when you get to larger brains, you get the folding, and the very small brains tend not to be folding. And so if you look at the fossil record for mammals, it seems pretty clear that the earliest mammals were very small. And so to my mind, it makes sense that the earliest mammals therefore were lissencephalic, and as they got larger in the various lineages, they became folded. And I agree with you that probably the mechanisms, or the active mechanisms or the constraints or whatever it is that generates folding in a size-dependent way, they were probably there in the beginning, but they don't manifest if the brain is small, right?
Yeah, exactly.
Yeah.
So we could say that there is a conservation in the developmental tools that then give rise to different adult structure, more expanded or bigger or smaller, but the tools are the same, right?
Yeah, I think that's true. I'm a little nervous about having latent things that are there but not really, right? But the potential for them exists because, then you'd very quickly get to, there's a history of like, what is it called? Preformation theory, right? Where you have like in the head of the sperm, you have the whole embryo, the whole adult is already preformed, but it's hidden and it's latent, but it will develop over time and then you can see it. I'm nervous about things like that because the truth is, I think, a lot of things are there and they arise through the interactions. There's a wonderful book, a very old book now, but by Susan Oyama that is called The Ontogeny of Information, and that's part of what I took from that book is that when you debate about is it a blueprint, is it a Bauplan, what exists in the embryo before it has developed. And she says basically not much exists, but the information to specify the interactions that will generate all these other things, that's already there. And then it gets combined with environmental inputs and so forth. But anyway, the second part of your question, I'm not sure I followed very well.
No, I think that makes it. But I just wanted to add that I have seen some recent work by some paleontologists that are looking to making endocasts, but with new techniques that look pretty good. And this could shed some light, not only in mammal evolution, but in any vertebrate lineage evolution, to know a bit more of these ancestral brains that we are trying to reconstruct in the book, right?
Sure.
So I'm curious also to see that.
Yeah, maybe you can share those references in the chat.
Yeah, I will have a look and I will send it.
If people are interested.
But I understand, Georg, that when there is no monophilic relationship between the different species, you do not accept that as being the same process. You think it's convergency producing a similar process every time, but it will be analogy actually, not similarity. Well, how are these concepts okay?
Yeah, so that's where I come back to.
You have a monophilic relationship, then if it's a species outside the monophilic tree, then you have to speak of convergency, and then even if the same gene, the same molecule, in the same cells or whatever, it must be different, non-homologous, or not.
So for me, that's why, as a grad student, I struggled with this problem of levels of biology, and you can have homology at one level without having homology at the other. And so what I think the question was about, you can have developmental mechanisms that are there in an embryo, like proliferation, or the fact that you can't stretch a cell infinitely, or things like that, or that these mechanisms, these conserved mechanisms, when you now increase the size of the animal in different lineages, they will lead to, the results are convergent, but the cellular molecular mechanisms that led to them are very ancient. That's how I would answer that question.
So I was wondering whether it's possible to define homology depending on the level or something. I'm thinking of someone that wants to record in an area in the Pratt and then find the same area that does the same thing or that has the same connections or that has the same something same. But if you just say they are homologous, those areas or not, you don't know whether they are similar in the way you are interested. So I don't know if there are other terms that could be used to define these homologies.
Yeah, so, for me, these were questions that, so for example, there are crickets, right? They have these courtship songs that the male crickets perform. And if you look at a lot of different cricket species, some use they rub their wings together, some use their legs to rub against the wings. So the mechanisms, how they produce that song, has changed over evolution, but I'm pretty sure all the intermediate crickets had a song, right? It's just that they changed how they were doing the song, the underlying mechanism. So in that case, I would say courtship song is a homologous character across all these different crickets. When you ask the mechanism and which mechanism is used in the behavior in different species, that's when you have independent evolution. Like maybe you have two species that independently started using their legs to rub against the wing when all the others were using just the wings rubbing together. So that's how I think of it. But yeah, it gets very confusing, and I'm not sure it's terribly productive.
But that leads us to the issue you mentioned in some publications years ago, and but you also put it in the book, that one cannot directly move from embryonic homology into adult homology. So you establish there a frontier, which I must say I never understood, because for me, anything you have in the adult, it comes from the embryo. So somehow it comes from the embryo. So I don't understand why. I don't understand your barrier, actually. I would like you to explain your idea on that.
I think that the cases I worry about where homologous characters can come from non-homologous embryonic precursor regions are so rare that I don't really want to make a big deal out of it anymore at this point. So in a way,
awareness in this field, what it needs to be interpreted is meaning that some of our assumptions are wrong. So if things are very weird, usually it's because our assumptions are wrong. So we need to analyze very carefully our assumptions in these cases, because maybe something that we assume that we know, maybe we don't know it properly. For instance, I remember you mentioned as one special case whether the neural tube has a cavity or not, depending on the species. No, for me, that is a problem of your assumption of what is a cavity. Because you may think that I don't see a cavity when I look at the microscope, but maybe if you look with the electron microscope, then you see a much smaller cavity that you can see with the electron microscope. You see what I mean? So whenever one finds one of these strange items of embryology that seem to make it impossible that you move into adult, you have to accept that the higher truth is that the development produces the adult. That means that whatever you have in the embryo is produced in the adult. So there must be a way. There must be a way out. And usually, I think is that we have wrong assumptions. We have to analyze our assumptions. That's my recommendation, at least.
I don't want to.
The concept of potency is also very important. You mentioned it before, because the embryonic cells have higher potency than they will have when they are adult. So they can do things in the embryo that they would never do in an adult. So that's another thing to keep in mind, that the embryo is not a small adult. An embryo is an embryo. And then it becomes an adult, which is losing a lot of properties. In order to become an adult, you lose many embryonic properties.
There's another kind of thing, though, that goes on. I mean, you mentioned about, cells can't stretch. Well, in the last 15 minutes of conversation, the role of the fact that, at least in mammals, I don't think it's been studied elsewhere, that the bulk of axon elongation is stretch, not growth. So if you look at the number of, the total volume of axons and myelin stuff in the brain, that how much of that is contributed by things growing from one thing to another as following a precursor then being stretched, it's almost all stretch. So if you start to look at that role of elasticity, it just shows up everywhere. And this is another kind of an explanation to look at, which is sort of a web of things that are produced by a very common basic cellular mechanism. So David Van Essen has his account of why cortical folds are the production of stretch. But that can be related to a computational issue because it solves one aspect of the save wire problem to minimize connection volume. So the simple fact that there's an elastic force created by stretch, makes the folds, makes the things efficient, makes the wiring of the cortex efficient. But back in the retina where I used to work, a whole bunch of people found that a lot of the receptive fields of single retinal neurons were elongated. That's where elongated receptive fields come from. And it turns out that I'm not sure about that latter part, but they were also generated by stretch. So you take a retina, put radial force on it, and everything becomes radially elongated and not the circularly symmetric field. So here's another case where the nature of information processing is changed by the elastic properties of the tissue. The production of the diminished periphery is also stretch. I could go on, but I won't. I mean, so if you start pick a really kind of good cellular property and then follow its tendrils out, you start to find a whole lot of things that can be brought under a single explanatory web in this case. And that's different from starting from a single developmental gene expression or an adult computational thing. I think we should consider as a class of explanation more often than this is done.
George, you want to add something?
Do I want to add something?
George? Yeah, it seemed to me
it
that
was
you wanted to...
very eloquent and makes good sense. I was thinking mostly when I think of cortical folding, what causes the folding, I do think it is buckling forces have a lot to do with it, and that's what I was trying to say about elasticity. It's like compression and stretch are real thing, and I think to me that's been really fascinating to watch in developmental biology, including cancer biology. The mechanical effects on cell behavior are just really interesting. And you mentioned the stretch. Yeah, a lot of axons are stretched. They grow in between, right? There's a stretch induced growth.
Yeah.
But but it's they don't just become thinner, the axons.
So I will read Paul's questions, unless you want to try again, or Paul, you want to try to, I don't know.
I can try. I don't know if it's any better.
It seems.
So my question is about the slopes that Barbara shown in different structures. They vary with respect to brain size, and there's this very strikingly different slope for neocortex, right? Wondering the
No, we lost you. We lost you again. Sorry.
But
Lose you.
hear it?
Yeah. I'll read, if you want, I can read the question. Yeah. So this is a question for Barb and George. You show how the slopes of brain differ from different structures. Can you comment on whether we might be able to infer functional transition from when those slopes change in phylogeny? Examples? He's asking for examples.
Well, first off, the cortex and the forebrain and cerebellum are rather similar in their slopes. And I was originally taught by Glenn that I should not think that the fish just made their cerebellar structures large, but that they also did their made their forebrain. And with Christine and with Carrie Opak and all those guys, it looks like it's not too easy to find inflections in slopes on the forebrain across all the animals been studied has pretty much the same higher slope, and I don't know if it would be really profitable to go into one of those allometric 50 million citations down. Is it really 0.06 or 0.04 or something? It's always higher. And it's not a very good strategy.
Yeah, so I don't think I know of any really good examples of changes in the scaling rules. I think like some of the best examples for changes in scaling rules come from Susanna's work on, the density and the number of neurons, right? Where you have this one set of scaling rules for rodents and a different one for primates. And you see that within the birds as well. So for me, that's been really eye-opening to see that scaling rules can change. Now, related to Barb's, the different slopes for different structures, when I was working on birds, it occurred to me that you could have some avian, say a parrot ancestor that said, hey, I'm going to mosaically make my tectum larger by delaying neurogenesis there, prolonging neurogenesis there. That would make that bird have a larger tectum. If you now say, well, this species then flourished and radiated and generated all sorts of other species that vary in brain size, well, that if you really believe that the variation, the scaling rules come about because of stretching and compressing these neurogenetic schedules, well, the ancestor had modified the plan, right? Mm-hmm. And so the consequences are going to be different in that particular group of, parrots in this case. Mm-hmm. They would have a different slope for the tectum. They would presumably have a higher slope for the tectum than the other birds. So I don't have good examples, but I'm sure that exists out there somewhere, because some animals do make parts of their brain larger by delaying neurogenesis, because that's a really good way to do it. And going back to computational things, although I'm not sure I entirely agree what computation means here, but I used to think about this. It's if you want to make a brain region larger in development, as it develops, you can delay the end of neurogenesis, or you can just expand the precursor region, right? But when you expand the precursor region, the neighbors must get smaller, right? You have to take it. Well, not necessarily, maybe, but yes. Simple-minded way of thinking about it is they must get smaller. So a really good way of making brain regions larger is to delay the end of neurogenesis. The cost is you have to delay maturation. So this is, I think, just in general, I think is a nice way of thinking about evolutionary changes and development, that there are costs and benefits to these various ways of changing development. And I learned that from Barbara. I don't know if I learned the lesson well, but
many of these basic developmental mechanisms, they are said to be heterochronic. That means that they are occurring with different beginnings and ends, and they can be modulated genetically so that the disproportions change. And recently I have been studying neurogenesis in the brain in comparing rat with chick and with the lizard, and we have found that the pattern in general is the same, but different parts of the brain, different areas of the brain, actually you can observe how they quantitatively change their sizes, the relative sizes, and even the morphogenesis. The shape of the tectum, for instance, is different in a bird as in a mammal, and that relates to the heterochronic differences you can see at early stages. So it's a correlation with the shaping because of this sort of playing around with the heterochronic processes. So the processes by themselves are not fixed. They can be moved around. And that's a good system for producing evolution, of course, at least adaptation, producing adaptation. Whether you obtain novelty in that way, that's a different story. But at least making bigger or smaller given parts, that can be done easily, I think.
So
novelty is if you invent in the patterning mechanism a new area. So instead of having a venula and then thalamus beneath, you invent something in between, then you have invented something new.
Okay, it's almost time, but Paul has a very good question for finishing this session and the whole series. That is, what's next? What should future generations of scientists be doing?
I think if I was a young person, I would get into that game of the single cell RNA-seq stuff, but couple it with connectional information them as best I can, or with some physiological aspect. Try to work your way from there to function. And I think that's very promising.
In which
Kind of
animal?
scary. It almost doesn't matter which animal, because you're going to be, there are going to be lots of people working on different animals, I think.
Barbara?
If you're in a hurry, it might be nice to compare I've always thought comparing different strains of mice is something that not enough people are doing, and because you have amazing tools and you have behavioral differences, why not? Sometimes you even know the history of how these strains were generated.
Mm-hmm.
Do you want to add something, Barbara or Luis?
Did you ask me to comment on that? I don't want to. I just want to do one thing that I've observed. So I'm being an editor now for over 20 years of this explicitly multidisciplinary area. I watched this sort of dismaying thing happen, which is that all the sub areas are going farther and farther apart, never co-referencing each other. The whole almost identical fields can appear in something you call self-psychology, something you call self-behavioral ecology, and another computational, and they will not be aware of the fact that these entire separate literatures actually exist. I think every sort of graduate student and every graduate committee should make a new role, which is kind of designated translator function. So you pick your area, and you're responsible for sort of at least following two of them, so that these things. I'm really quite worried about what's happening because, the utter waste of time and ridiculous debates and things about things that shouldn't be debated are just seeing over and over again. And I don't know how to solve it in general, but maybe some kind of a little bit of a change in how we think about graduate and postdoctoral education, which is to, instead of it being this, I'm going to keep track of everything, which you cannot do, maybe everyone should keep track of their own field and some other thing, and then make a consortium of these people so that we can actually move forward in some kind of coherent way, because otherwise it's getting depressing. So we've got to figure out some way to keep things together, not just cells, not just behavioral function, not just computation, but you have to do all of them for brain. You just can't, you must do that. So you got to figure out a way.
Luis, what would you do your PhD on if you could?
You're speaking to me?
Yeah.
I didn't understand your question.
So it was posed questions. What would you work on if you had to decide now?
How would I work?
Yeah.
I don't get what you mean.
Well, if you had to start again your career, what would you focus on? With the new tools and the.
I think we need much more work on the connection of developmental processes and the adult structure. Think that evolution, where it occurs, is in the production of a new ovule. Once you have the zygote, that's where you have the new genome. And that's all where you contain in potency, everything is going to happen. So all the changes between the zygotic stage and the adult, everything is the process of producing whatever the genomic change has introduced. That's the general idea. So that means tells you the importance of developmental analysis. Because with the ovules only and with the spermatozoids, we don't do anything, obviously. So we need to transfer the information from the zygotic state to the adult, a much more complex state, millions of times more complex. So that is a real difficulty of understanding evolution and how complexity arises and all this sort of thing. The secret is in the embryos. So if you don't study the embryos, you only can speculate. That's a problem with functional speculation. If you don't have a basis for speculation, then it's free speculation, which can be very entertaining and very important because it makes you think and it makes you look. And so I don't deprecate speculation. I like to speculate myself, but we need to know that the problem is how do we transfer the genomic information to the adult brain? That is the big question. That's a real big question. Once we
Can I
yeah.
I wanted to come back to something Barb said because I was just thinking about it. I think, this idea of a translator and reading outside your main field. It's not just if you don't do it, it's not just that if you don't do it, it's a waste of time. I think it can be an enormous benefit to have outside information that the people in your little area don't have. So I'm thinking, for example, I hope this is fair to say, I think it's fair to say that Glenn Northcutt, one of his main secrets of success was that he was reading the general morphological literature and the phylogenetic systematics that wasn't being discussed inside neuro, and then he brought all of that over into neuro, and you could say, oh, he didn't come up with it himself. It was like, well, there are a lot of smart people out there, a lot of good ideas, and if you bring them together into a new area, good for you. I think, we don't need to reinvent the wheel, and I think that's what you were saying. We don't need everyone to reinvent the wheel all the time. Some good scholarship.
This is more or less what Paul was suggesting in the chat. Read that which others in your field don't read.
Yeah.
Basically.
Yeah.
That was the actual one of the aims of this reading club, actually, because I am from computational neuroscience, Raquel is from systems, Paul is from many different fields. So bringing people from different fields together. So maybe we can finish with this. I will tell Raquel and Alex to turn on their cameras to say goodbye. And I would like to really thank you, Barbara, George, and also Luis and Paul that have been here all the sessions, 7 sessions. It's been a pleasure for us. It's been, we've learned a lot, as I said in the beginning. It's been amazing. So we will have to think of another book to discuss.
Thank you so much.
Thank you so much to all of you. It was amazing this journey.
Bye.
Thank you.
Bye.